You like?
I think so.
Okay, awesome. Hello, my science family. I am so excited to you guys back for another awesome day of exploring the human body. So our first day on Monday, we sort of looked about the outer body. We looked at hands and how muscles and tendons work together to allow us to move our hands, which was amazing. And then yesterday we went inside our body, but we looked at a really big picture of our body. We looked at the digestive tract, and that was amazing because like the comments were like, ew, disgusting, I can't watch, to like, oh, cool, this is amazing. So I loved it. Even one of my kids was like, nope, can't do it. And one of my other kids was like, yes, this is amazing, I cannot wait. So I love getting those reactions with science. And today we're going to go even deeper into our body, and we're going to look at human cells, so the building blocks of the body. And tomorrow we're actually going to extract something from inside cells. We're going to extract DNA. And one of the things you need to do that is going to be alcohol. And I need you guys to tell your moms and dads to go put some in the freezer. So we want it cold. If you don't have alcohol that looks like this inside of your medicine cabinet, you could probably use some hand sanitizer. We won't be using a ton, so it shouldn't like impact that. But ideally something with really high alcohol concentration you're gonna need, and we need to put that in the freezer now so that it'll be nice and cold tomorrow when we extract our DNA. But today we're gonna look at cells, which is amazing, and we're gonna do it by making a model of a cell. And you guys, we've talked about we want to just like gather whatever. So I have like all this crazy stuff. I've got like, I think I'm going to use like this my cell bottom. I've got markers. I've got like spaghetti, pipe cleaners and straws and tape and rubber bands. I got like baking cups for cupcakes. We've got, you know, markers, whatever. I just put a whole bunch of so that when I make the cell, I can be really creative and think about what I want each part of my cell to be. And the most important part of our cell is going to be our supercell action. So you guys also want to think about, what am I going to make my cell do that's going to make it extra special? And we can share it in Zoom. So I'm really excited to see the comments now. If you want to make sure you never miss any of our science that's been happening, we are doing currently 6 weeks of science, but it's going to extend for however long school closures work. So we will keep producing some awesome science for you guys and sort of making a nice fun part to your day that you can experience with us. We do have a couple of things that we are going to look at today because we have some ongoing experiments. If you're just joining us, we have been, science takes time. So we have some little projects that even if you can't do it at home, you can sort of watch it in action with us. And one of those projects is making mummies. So in week 1, we learned about osmosis and how to make a mummy. And we started that process, but the mummies take a little bit of time to create. So we are slowly changing our mummy salt every single week. And I'm going to change it for both of my girls today. We've got 2 different mummies. All you need to change your mummy salt, if you're doing this at home, is salt, baking soda, and a new Ziploc bag. If you are just joining us, you've like just come recently, you don't actually need to like just watch it. You could start your own mummy. It starts the exact same way we're about to do it. So you could start it and you'll just be a little bit further behind on what it looks like. But it only takes about 4 or 5 weeks to make a mummy. So you can still get all of this done. All right, so when we make our mummies, every time we change the salt, you want to bring out your little item, whatever it is. You want to scrunch off as much salt. Isabella did a lemon, and it's like definitely you can see it losing some of the weight. Now hers started at 2 ounces. And then it went to 1 3/8 of an ounce last week. And this week, let's put it in ounces, it is 1 1/4. So it's still losing some weight, not as much, because 1 1/4 of an ounce is the same as 1 2/8 of an ounce. So it only lost 1/8 of an ounce, but that's okay. Maybe we're getting most of the water out. Now that I've weighed it, I'm going to take my little bag, I'm going to add a bunch of salt in. And I'll also add some baking soda in, and that is what the Egyptians kind of basically used to make a thing called matron, and they were stuffing humans, their leaders. They made mummies out of so that they could commemorate their leaders and have somebody to go visit. So I've been placing it sort of cut side down so that it can get all that good stuff in the salt, and then I just like to fill it up with salt. This project takes a lot of salt, which is awesome. And so once I have that, I'm just going to zip the bag up, and I'm going to store it again in somewhere that's sort of cool and dark. We don't want the sun. All right, so we saw what a lemon looks like. This part, you're just going to want to throw away this extra salt. We don't want to use that. And a side note, if you are doing a meat, some people might be doing like chicken legs, totally fine. Just make sure that when you're touching it, you actually use gloves. Fruits, I'm not too worried about bacteria on. And even though if you did chicken, you washed it down with alcohol before, we want to be really careful. So that's something we want to do in science as much as possible. This is George's banana. And we didn't change it last week because we had just changed it. We had to change our banana a whole bunch of times because there was so much water coming out. And you can actually see how like little her banana has become. And I don't remember off the top of my head how much hers weighed. We have to go look at her book to see that. But right now it's 7/8 of an ounce. It's not even a full ounce at the moment. So we can record that with her later. And I'm going to do the same thing. So I'm going to fill up this bag with a whole bunch of salt. And the water, remember with osmosis, it always seeks to sort of dilute. So the water is going to go out of the banana where there's a lot of water and into the salt where there's not much water. And so we get sort of this dried out banana during that. And we're going to add a little baking soda. You don't have to add too much. And then we're going to fill it over the top with some more salt. And these guys will be good to go for another week, or, you know, if you forget about it, a week and a half, it's not a big deal. You don't have to do it exactly on time. And again, this salt, this old salt, and you can even see the difference between the color and sort of like this one's crackly because it's got so much liquid in it. This salt we're going to throw away. So that salt is now dead to us. And we have our two new mummies with our new salt. All right. The other project that we have been working on that we started yesterday with our digestion was we wanted to look at enzymes. And there's like sort of three big parts of food. If you remember, there's fats, there's carbohydrates, and there's proteins. And our bodies have to break that down. And they do that through enzymes, which are basically a pair of scissors. So these little scissors, these enzymes go through, maybe even like a Pac-Man, and they go cut up all of those either proteins, or they cut up all the carbohydrates, or they cut up all of the fats. And they're very specific. They only like to cut one type of thing. And interestingly, the pineapple plant has an enzyme in it that chews up, it cuts up animal protein, which is sort of interesting if you think about it. Maybe it evolved that way because animals loved the pineapple and it made actually their mouths hurt. In fact, George last night ate so much pineapple because she loves it, and then was near tears and possibly in tears because her mouth hurt so much because the little scissors were in her mouth chopping up that animal protein. So what we did is we set up an experiment to look at what happens. And we did that with clear gelatin. Gelatin is just animal protein and water. And we looked at actually a meat tenderizer. Meat tenderizer is pure bromelain, which is just an enzyme, straight scissors. And then we cook the pineapple, because when we cook the pineapple, it actually breaks our scissors apart. So you sort of have like, instead of having like little scissors like this, chop, chop, you have like two things that are just like, they can't do anything. They're just like hanging out. And then we have raw pineapple, which also has bromelain, which is that enzyme that eats animal proteins and has the scissors. So we're going to keep this project over the weekend too, but I want to take a look at what's happening. So in the meat tenderizer, there's so much liquid in here. If you just sort of look at about, and all of that liquid from the protein in the chopped up and it having to release the liquid it was holding on to. So this one worked really well. There's a whole lot of water. And then if we look at the other one that has an active enzyme, which is just the pure pineapple, it's raw, it hasn't been cooked. So those are intact. You can see all of this liquid down here. That liquid is from them chopping up those Have lots of liquid in this one too. And of course, we always want to check, like, if I just put something on top of gelatin, will it always turn into a liquid? What's going on? So what we did is we actually did raw pineapple, or not raw pineapple, sorry, cooked pineapple. And the cooked pineapple is the one where the scissors, we broke the scissors so they can't cut anything. And if you look at it, there's no liquid on here. So they haven't chopped through those proteins to be able to release the water. And again, if I look at the pineapple that was raw, that has those scissors intact, I've got a whole bunch of liquid. So we're going to leave this experiment running over the weekend. I'm actually going to cover them so we don't get our water that evaporates. And we'll see what it looks like. We can check in on it actually tomorrow when we do DNA and Friday. And we'll just keep leaving it to see like how much can it eat through. So those are our little ongoing science experiments that we've got going on. And now, without further ado, let's do our shout outs so that we can see who is with us. Because I love seeing who gets to come with us every day. It's going to be so exciting. Who is with us today?
All right. Today, first up, all the way back up here, we have a pretty active chat feed.
Yes.
We have Rachel Floyd. She's here.
Hello, Rachel.
And you know what?
How are
She
you?
said, I love your channel with 7 exclamation marks.
Yes. Which also hopefully means you love science. Woo! Which means I'm doing my job. Yes. Makes me so happy. I love science, if you didn't notice.
Then we have Naomi. She's here again.
Hello, Naomi. Oh, it was so good to see you in Zoom yesterday. I'm so excited about all of our new friends.
And Megan and Taylor, the Hertz.
Hello, Megan and Taylor.
Sure.
Where's Jacob?
Chloe and Calvin are here.
Hello,
But where's
Chloe and
Amelia?
Calvin, and where's Amelia? We're missing some siblings today.
George and Henry say hi.
Hello, George and Henry. It's good to see you guys again. Also, I love that we know the siblings who are missing. Like, these are people we didn't know before. I love that we're creating this fun little community.
And Connor says hi.
Hi, Connor.
And Julie, our friend Julie, says, Happy April Fool's Day. Maybe she's got a trick in store for us. I don't know.
Maybe. Ooh, I did not trick my kids yet. Interesting.
They have plans for you.
Our kids have plans for me?
At least one of them does.
Ooh.
Then we have the good kids, Ben, Sam, Ellie, and John.
Hello, Ben, Sam, Ellie, and John. And you have the best. Oh my gosh. Those kids are good. They're really good kids. Every day. It never gets old for me. It might get old for you guys, but it doesn't get old for me.
Abel says hi.
Hello, Abel. Oh, I'm excited that you're joining us.
And Erin says hi.
Hello, Erin.
And then Venetia.
Venetia.
I got it. Hey,
I saw
Venetia.
him. I saw the tears cranking in Evan's head. And there's only one name I have trouble pronouncing.
I know. I'm getting it. It'll be clear. Then we have somebody asked, what is the best way to dispose of the salt?
The best way to dispose of the salt, actually, if you're gardening, would be to find a spot in your garden where you have weeds and you do not want them to grow. Like, you don't want anything to grow. So maybe you want to install like a walkway there. You could salt the earth there. That will like kill everything, but nothing will grow there. So don't like be like, I'm going to salt the earth and then plant tomatoes. That wouldn't work. That would be an environmental way to do it. Or you could just put it in your trash. Or if you compost, ooh, that might be bad for the worms. Don't put it in the compost. No. Worms, that might be too salty. So I would think maybe the trash, or if there's a spot that you're trying to kill a whole bunch of weeds and you don't want to grow anything there. Ever. Well, not ever. It's going to go away. Rain will come, but you could salt. It's called salting the earth and it will kill whatever is there. So like we might take this salt out into our garden where we are battling blackberry bushes and put it sort of on the little walkways that we're creating. And it will kill it for us, which is handy.
I don't know if I agree.
I've been saying no. I am the black thumb. I kill everything in the garden, so maybe that's why.
She's very good at this part. We have somebody else said that they love their channels. Amelia, the Lindbergh said so.
Yay! And that must mean Amelia's here.
Yeah. So, and Julie mentioned that Rome using salt was how Rome destroyed Carthage.
Oh,
they salted their. That's the original.
That's where it comes from?
Yeah.
Julie, you taught me something today. And it's about history, which I'm terrible at. Oh, I'm so excited. The Romans salted the earth to basically starve out the who?
The Carthaginians.
The Carthaginians. Ooh, that's cool. Look at that. Oh my gosh, amazing.
All right.
All right.
So we have a couple more.
Oh,
we have Raiden says hi.
Hi, Raiden!
Yeah.
Raiden was an astronaut in Zoom yesterday. It was amazing. He had a cool background.
Oh,
I loved
cool.
it. Yeah, it was cool.
Yeah, definitely explore the backgrounds in Zoom. Jackson. Hey, Jackson.
Hi, Jackson. Welcome back.
And it looks like just Ruby, no life.
Oh, well, I'm excited that Ruby's here.
Oh, Jacob's here now.
Yay! Yay, Jacob! Welcome.
And Amelia has a headache. That's why she's not here.
Oh, I'm sorry. I hope Amelia feels better.
And Vinatia has not yet pranked her mom, but it's on the plan.
Okay. I like it. Are we ready?
We have one more.
Oh, one more.
Julie said that you could also dilute the salt and probably just run it down the drain.
Yes, you could totally. That's totally a way that you could do it if you wanted to.
I
thought we were gonna like maybe use it in something else, and we should not use it in anything else. But yeah, diluting it and running it down the drain.
If you're on a
would work.
septic, though, that might destroy some of the bacteria that help your septic system function. So be careful with that too.
Yeah, I mean, this is not like a huge amount of salt either. I can't kill that many plants with it. Evan's over there like rolling his eyes at me, but I'm like, I think it seems like a good idea. Maybe not. We'll find out. Alrighty, let's make a cell, and you can make it on anything you want. So if you have like a piece of cardboard, if you have a paper plate, if you want to use like a Tupperware, you could be using food items. Lots of people make things out of food to make it more fun. You could use like anything in your craft drawer. I just have like all of this stuff. It's going to be fun, and we're going to see what we do with it. So first, we're going to make sort of a model of our cell, and then I'm going to add some sort of superhero power to our cell that's going to make our cell stand out. Now, here's the thing with cells. Like, our body is made up of all different types of cells. So they're like the little tiny building blocks of cells, and there are more than 30 trillion cells in our body, which is like crazy. So when I say this next thing about how many cells die, it's actually not that many compared to 30 trillion, but 50 billion cells in your body die every single day. 50 billion, that's so cool. And it only takes like a day for a lot of these cells to make new cells. So it's not actually a big problem that we're losing 50 billion cells, especially because we're made up of 30 trillion cells, which is such a big number. It like blows my mind. It's so hard to think about. So much. 8 billion people live on the planet. So like that's like 5 cells dying for every person on the planet that lives in your own body, in everybody's body every day. And cells, they are that building block of life. So everything that's living has cells. Like plants have cells, we have cells, bacteria are just single cells. So they're cells. I mean, everything that's living is a cell. And even some things that are dead are cells. Like my hair is made up of cells, but they're dead cells. Or this table is wood and it's made up of cells, but it's dead plant cells. But then there's things that are just not made up of cells. Like, well, cardboard is actually made of plants. I was going to use cardboard. It's made of plants. So there's plant cells in there. My scissors, however, are metal, and metal is not from a living thing. So that's actually not made up of cells. Or the plastic on here is not made up of a living thing. So that's not made up of a cell. So there's some things that are made up of cells and some things that aren't. And if it was living, then it's still made up of cells, which is really cool. Cells are all different sizes. So there are cells, they're all really small. None of them can we see with our naked eye. So we have to use a microscope to look. But even then, there's a cell in our body that's 4.5 feet long. What? How can we not see a cell that's 4.5 feet long in our body? Well, it's really long, but it's super skinny. So it's like a micron thick. And to like sort of give you a comparison, if you pull out like a single strand of hair, which you guys can't see because it's so thin, but if you did this on your own head, that single strand of hair is 75 microns thick. So 75 of these cells could be bundled together and it'd be as thick as your hair. So it's still really, really thin, which makes us not be able to see it. And those cells are actually our motor cells that allow sort of the signal from our brain to go tell our foot to wiggle the toes. So we have cells like that. We have cells that group together and send out signals to each other that are like, everybody pump, everybody pump. And they do it together. They're like, woo, together, woo. And they do it on a beat. The beat sounds like this. and it's our heartbeat. Our heart is made up of cells. It's a whole bunch of cells that got together and organized themselves to do the same thing at the same time. And we get this awesome heartbeat. We have cells that create our muscles. We have cells that create our skin. Everything in our bodies is made up of cells, either living or dead, which is amazing. None of them can we see with our naked eye, but we are going to blow one up and check it out today. And all of the cells in our body, from the heart cells to the motor cells to our immune system cells that have a memory, literally have a memory to remember, like, oh, hey, Dr. Erica, you had this cold last year? That's right. I remember what that cold looks like. Or a memory of, oh, hey, Dr. Erica, remember when you were 4 and you got chickenpox? I'm never going to forget it, which is really, really cool. So all of those cells have the same structure. So they all have a cell wall. So the first thing we're going to have is have to figure out a cell wall. I'm going to use this as my cell wall, and I'm actually going to cut a hole in it so that I can like have like a cool sort of like 3-dimensional cell. So whatever you want as your cell wall, think about what you're going to use, grab it out, and let's get it ready to make our awesome cell model. All right, so I'm going to cut sort of around this. Okay, right here. So I have a window into my cell. And the cell wall is really important. It lets things in and out. There's gatekeepers that say, you know, we need more water. We don't need more water. We need more maybe calcium ions. We don't need more calcium ions. Hey, we've got these things we made that you might want out there. So even these cell walls, mine doesn't look like anything could get through it. But there's actually all of these pores in there that lets things get in and out. And I'm going to just sort of, I think I'm just going to glue it like this so that, whoa, I've melted a glue stick to my glue gun so that it's got a little bit better structure as I'm building. So hopefully you're grabbing out whatever you're going to decide you want to use for your cell walls. That's going to give your cell the structure it needs.
So we have a question.
Yeah.
Doesn't an animal cell not have a cell wall? I thought it was just plant cells that had walls.
Ooh, we all have cell walls, but they look different. So plant cells have a really rigid cell wall. Animal cells have a lipid bilayer as their cell wall. So we have a bunch of fats that go around. So it makes it like wobbly. This would be a fantastic plant cell model because it's really rigid and structured. Animal cells still have that cell wall. It's just like much more like gooey. Yeah, like, you know, those like little squishy toys we have and you can like squish them and they can do all the sorts of things. Animal cell walls are much more like that. They're much more fluid. This one does not. It's a great point out. This one does not have that, but that's okay. We do our best. All right. So let's look inside of our cells. So yesterday we looked at our digestive tract and we looked at how our digestive tract takes energy that we put into our body and turns it, or food that we put into our body and turns it into energy. The cell has to do the same thing. It has to take nutrients it might find around the cell and turn that into energy so the cell factory can keep on pumping. And that thing is called mitochondria. So it's sort of like the little digestive tract. And so we just think about, what do I want my digestive tract to look like? I don't know. I think I might use these. I have bow tie noodles. I think I might make these my mitochondria because I think it's going to remind me that like I eat food to make energy and maybe like these guys eat nutrients to make energy for the cell. So I'm going to put some mitochondria into my cell. And we're just sort of having fun. So we don't have to be like worried about like, did I add too much or too little? We're just making really cool cells. So I'm going to add some little digestive tract so my cell can have lots of energy. And I probably need extra energy because my cell is going to have a really cool superpower.
And if you don't have a hot glue gun, you can tape it in. Yeah,
if you don't have a hot glue gun, you can use tape. If you don't want to use any of this stuff, you could just use markers and draw it. You could use markers to label things. All right, so I have lots of things that will remind me of how delicious it was, making me hungry just looking at these noodles about, I want to put some energy in my body and I've got some energy in my cell. Awesome. All right. So another thing that cells have is DNA. And this is the thing that we're going to extract from cells tomorrow. So you're going to extract it either from like a banana cell or a strawberry, some sort of soft fruit, or probably the second time you do it, maybe even your cheeks, which would be really cool because you would have your own DNA. That is so cool. So we need DNA, and DNA is like the storybook. It tells us who we are and what we look like and what we do. So for a cell, the DNA kind of tells it like, what is your mission in life? And so I'm like, oh, it's a storybook. I think I might make my DNA kind of look like a storybook. So obviously it's not looking exactly like what things really look like, but I'm more interested in it like reminding me. I'm gonna make a little booklet. Maybe it needs to be a thick booklet because DNA is really long. I'm gonna make it thick. We'll add some like thick pages to this like delightfully good read that we're gonna be up till midnight reading tonight. It's gonna be amazing.
Where is DNA located?
Ooh, great question. Yeah, DNA is located in the nucleus of our cell. So we don't actually have a nucleus yet, which means I can't really install my DNA. So DNA is kind of like all of the books that you might find in a library telling you, it's like your instruction guide. Oh, how do I make chicken soup? You go to your DNA to find out how you made chicken soup for your cell. And it's like maybe, oh, how do I decide what to do right here? And oh, let's go find the instruction book. So DNA is sort of like a little library of instruction books. And I'm going to maybe draw. DNA is made up of what we call a double helix. So it's two strands and they sort of twist as we go down. So maybe I'll draw that double helix to sort of remind me that DNA looks like this, and it's actually sort of the instruction manual. But our commenter on YouTube was right. Like, where are we going to put our DNA? I can't just like let the DNA float around my cell because then when something needs to access that information, it wouldn't know where to go. So I need to think about, before I install this, I need to make a nucleus, which is going to be sort of like a big blob in our cell that's got all of our DNA booklets in. So I do have a cup. I'm wondering if this is maybe better.
While you're brainstorming that, what would happen if you lost all your cells or just one of your cells?
Well, if we lost one of our cells, that wouldn't be a problem because we lose 50 billion every day. So that would not be a problem. Our cells would replicate and they would remake them and we would be totally fine. If we lost all of our cells, that would be a problem. Like, I mean, if I just like expanded myself and all of my cells all of a sudden decided to like run away from each other, I wouldn't be me anymore, which would be kind of sad. I would miss myself.
Dissolve and wash it.
Yeah, I mean, I think you would just like, you would turn to like dust, right? Dust is dead cells. So you would just, I mean, that's the whole thing. Dust to dust, ashes to ashes. Yeah. So you would turn into dust. They would
dry out. And then would turn to dust.
Yeah, and you wouldn't be alive anymore. It would be very sad because I would miss you. All right, so I think I'm going to use this as my nucleus. I'm just sort of debating. I have these cups too that are really cool.
What are the pink things in here?
Oh, I didn't put it on there. Those are vacuoles. Do we know much about vacuoles? Because I didn't put it on my thing to study because I'm not a biologist by trade.
I'll look it up.
Evan's going to look it up for us. Those are vacuoles. I'm surprised I didn't put it on there. Good job for you guys holding me accountable, though. I appreciate that. All right, so I think I'm going to use this. I can't decide, though. This looks super cool.
They're storage bubbles.
Storage bubbles. So they store gases?
They might store food or nutrients that a cell might need to survive.
Ooh.
And they can also store waste products. It's like the stomach.
It's like our stomach. That's pretty awesome. Here's also something really cool. As you guys are learning, I'm learning with you because I'm a physicist by trade. So when I did all of my research, I looked really hard at like, what does this water molecule do when this happens? And I shot things with lasers, which was really cool. But just because I have a PhD doesn't mean that I know everything about science. It just means that I really like to learn. So I'm totally okay saying I have no idea about something. There's nothing to be ashamed about not knowing something because you can just go look it up and you can learn it. And then you have that knowledge, which is really cool. All right, so this is going to be my nucleus. And our nucleus, the outer part of it actually has all of these holes in the membrane because things need to get in and out, right? If I want to access this data, then I need to have stuff get in and out. So maybe I'll actually even make some holes inside of this. This could be a disaster. Okay, wait, we're going to try again. You know what I'm going to do? I'm going to draw pretend holes on this one.
Ooh, what is a nucleus?
A nucleus is sort of like the center area of it. So it's the spot that has all of our information for our cells. And everything else in the cell is like really, to be honest, mostly cytoplasm, which is sort of like the goop that everything swims around in. And the nucleus is sort of like our information center. It's pretty dense, just like sort of an egg yolk is much more dense than the egg whites. I'm going to do it like this. I'm going to put it flipped over so that it can't get out.
I would amazing idea.
Yes.
We should totally do cells, but do it with slime next time. Like,
I actually thought about that yesterday, last night. I was like, we could do it with slime and put cool things into the slime. So then it was like all, but then those would be supplies that we didn't have. No, I have nothing against slime. I love slime, but not everybody has like enough glue or borax powder, or wants their kids, like slime can be really messy and sticky. All right, so this is going to be my, well, this is actually technically my nuclear envelope, but it's sort of like the outside of the nucleus. Where did I put my DNA? Ah, here's my DNA. I'm going to put my DNA book inside of here so that I've got a little book of DNA. I'm going to put it upside down so that I can have my nuclear membrane be sort of like hanging out. So also inside of this nucleus, other than the DNA, is our nucleolus. And that's sort of like a little factory inside of it. So super, super dense. And that sort of, it makes the ribosomes. So we haven't talked about ribosomes, but it makes these little guys that make other things. So it makes our makers, which is really cool. So I've got my DNA in here. I need the nucleolus that's going to make my makers. It's going to be super dense. I'm going to use this paint. This paint's super cool, right? The little paint thing, will it fit in there?
I don't know.
That could be like my little, oh, it's so pretty. Yes, that's going to be it. It's going to be, this is my little factory where I'm going to make my makers, and the pink things are going to be all my makers. Yes, love it. So you don't have to have a plan before. You can just be building with us right now and seeing what kind of things you can find around your house. All right, so this is my nucleolus where I'm going to build my little makers in. So it's like a factory that's going to churn out little makers, just like that.
There's no right answer. Whatever you build.
Yeah, there's no right or wrong answer. I mean, it's just, we're just creating. And the goal is that we learn something. You don't have to memorize these things. I mean, we'll leave that to like the teachers in 6th or 7th grade where you're like, you must know exactly what this does. We just want to get familiar with the vocabulary and what they do and have fun, because fun is really important. All right, I'm going to glue this down. And that is going to be basically my sort of cell brains are right there. So, all right, so I got that guy glued down, my cell brains in my library. And then I got to think about what else do I want? We could add our ribosomes. And that's the thing that's going to be made inside this nucleolus that are pink. So I want my ribosomes, actually, I think I want them to be pink. They're going to be pink. Oh, I don't have very many of these. Maybe Evan could go grab me some more. I got these perler beads. And I think I'm going to make my ribosomes be pink perler beads, because that'll remind me that they're made in this little spot inside the nucleus and that nucleolus. And these ribosomes, they are the little factory workers that are going to build proteins that my cell needs. So that's going to be amazing. Ooh, that one's orange. All right, so, and I'm going to put a lot because we got to have lots of ribosomes. And ribosomes are sometimes floating all around. I'm going to do the ones that are floating all around now. And sometimes they're actually at what I call the factory storeroom, which is the endoplasmic reticulum. And that's sort of the spot that holds a lot of the stuff you might need. Proteins. So proteins are built out of something called amino acids. And our endoplasmic reticulum is sort of like the store that these little makers are going to go to. So once we install that, we want a whole bunch of makers there, because if you're making something, you got to go to the store. But we're going to put some around the cell first. So I'm going to put in my little, these are going to be my little ribosomes. And they are going to be making all of the proteins that my cell needs to function properly. All right. My glue is like super duper stringy. This web. Maybe I'll just have like the glue strings be like my cytoplasm. Be like what looks like my cytoplasm. Like, look at this. It's got, maybe you can't see it. It's super stringy. I don't know what's wrong with my glue, but it is so stringy. All right. So here are a few of my little ribosome workers that are building my proteins, but we should make that storeroom. So the endoplasmic reticulum the storeroom is right by the nucleus because those little workers are going to need to learn what they need. They need to get the little recipe card to make each protein. So we're going to put the factory of storage maybe right here. We guys should think about what we want to make of. Like, I could put like boxes, like a shelf maybe. Or maybe I'll do like pipe cleaners because there's so many folds in the endoplasmic reticulum, so I can really store a lot of stuff. So maybe I'll like make like something wobbly out of these pipe cleaners, but then I'll put, they happen to match color. Well, they don't happen. I thought about that as I was pulling them out. Maybe I'll put these little boxes to remind me that's like sort of like a storage factory and it stores all of those things we need. I think I'm going to do that. All right, so I'm going to install my endoplasmic reticulum. And remember, I want it to be really close to the nucleus of my cell so that my little ribosomes can ask questions and get answers from that DNA storybook. That's really important. All right, let's see. So I'm going to glue it down here. And I'm just going to like sort of glue and bend. It's a little bit tricky to do upside down. But that will make sure that you guys have some time to work right alongside with me. All right. And again, if you just want to draw the pieces, you can draw the pieces. If you want to make it really big, you can make it really big. If you want to make it really small, you can make it really small. There's like, again, yeah, like Evan said, there is no right or wrong. We're just learning, and we just want to make a model to help us learn so that when we are at dinner maybe and we're talking about all the really cool science we did today, we can remember like, oh yeah, the DNA is the storybook. And, oh, right, the ribosomes are the little workers that are making things. That's really what we're going for. And bonus, when you guys get to middle school and you have to make this, you'll be like, I already made one of those. And you'll have all the ideas on how to make it. It won't feel like this onerous, hard project. It'll be like, I did that and it was fun. Let's do it again. Or at least hopefully. All right, here we go. I'm going to add a bunch of folds in my endoplasmic reticulum. I want it to be really big. We need to store a lot of goodies in our cells. So I'm going to add another piece here. We'll bring it back. I'm just realizing in my little quick sample that I made last week for the pictures, I also made my endoplasmic reticulum orange. Maybe I think of factories as being orange. I'm not sure why that would be. Seems odd. Alrighty. And remember, my endoplasmic reticulum is that storehouse, so I'm going to have some ribosomes there too. Although I've got to install my little boxes that I'm going to consider my like little storage boxes for my endoplasmic reticulum. All right, we're just going to glue this guy down here. Just like that. All right. Whoa. There we go. All right, so there is my endoplasmic reticulum, but I did want to install some of these like little cubes as a way to remind myself that the endoplasmic reticulum is the cell's storage area. So I'm actually curious if I can cut this down because this feels like really big if I put that in there. I'm gonna see if I can cut these. So be like, oh, perfect, I totally can. I'm gonna cut them one cube. I'll cut a cube into eighths. Make 8 little cubes. Just so I can remember that it stores all of the building blocks I need for proteins, which will be really handy. Oh, I'm really happy I can make that smaller. I think that would have been just too big before. All right, so we're going to create our organizational storage bins. Clearly, I like organizational storage bins in our house. And we're just going to stick those in that endoplasmic reticulum so it's just a reminder of like what it's going to look like and what its function is going to be. Because that's really what we want to do. We just want to sort of remember what are the parts of the cell. And if we can't remember the names, we want to remember, oh yeah, there's a spot that's like a storybook that tells the cell what to do. And there's things that make building blocks for the other things. There's things that give directions. There's things that store. And then we can learn those other things the next time we see them. Because a lot of learning happens not when you see it once, but when you see it 2 times or 3 times or 4 times. So this is a great way to sort of prep yourself for the next time you are learning about cell structure. Because I probably won't remember about the Romans and salting the earth later, but if somebody tells me that again, I'll totally remember it. All right, so here's my little endoplasmic reticulum with storage blocks. And remember, the ribosomes go there to get the stuff they need to build those proteins. So we do want some of our little pink ribosome makers to be at that endoplasmic reticulum because they got to go to the store and get what they need. That's really important. So I'm going to get some of these pink ones out. And they're going to get glued right in here. And woo, it's really hard to glue upside down.
Sorry for any jostling video. I'm trying to move 3 screens. Definitely let us know if you have any questions.
Yeah.
We're building.
I'm super excited. If you're not joining us in Zoom, you should give us ideas of what you are making stuff out of in the comments.
Yeah, the Zoom classroom is open.
Evan says the Zoom classroom is open. We're not really taking any things from the Zoom classroom right now, so I won't be able to see your stuff quite yet in the Zoom classroom, but we will be there in a little bit. And if you're a patron, you have that access to the Zoom classroom. If you're not, you should totally join us. It's pay what you can. So, you know, as little as $1 a week, if that's what you can afford to come do science with us, is amazing. Tell all your friends, because the more the merrier. It helps us ensure that our programs can continue to grow. And yeah, I mean, there's no reason why we can't have like 10 billion. Okay, 10 billion people would not work because that would be like everybody on the planet, more than everybody on the planet. We'd have to like enlist some aliens from outer space to make that happen. But that would like totally make my day if we had like so many people watching us do science. All right, so I have my little ribosomes in the endoplasmic reticulum so they can make their stuff. So if I look at my little thing, we've got to think about other things that we want to make. And we haven't made our Golgi apparatus. So the Golgi apparatus is sort of like a post office. So these little guys, these ribosomes, they can build the proteins, but then we need to package up those proteins and send them to another part of the cell that might need them. So it's sort of like a post office. And it usually goes like right around here by our endoplasmic reticulum and really close to the nucleus. And that's because we have all of the building blocks to make the proteins here, and we've got a lot of the ribosomes to make the stuff here. And then we want to be able to ship them off really fast, you know, express mail. I think I'm going to use straws. I feel like I was drawing straws because the Golgi apparatus is blue inside my little printout. But I'm also kind of thinking these straws, like, you know, when you go to a bank and you can put like the money in the thing and you press and it goes shoop up. The straws are kind of reminding me of that right now, of like that little way to shoop things like places. And it sort of is going to help me remember that this is the post office. So we're going to like package up those proteins and some fats together, and then it'll throw it out to other parts of the cells. So I'm going to use this as and I'm going to cut it down into smaller pieces. Whoa. Okay, maybe that one is not going to be used because it went off our table, but that's okay. And the Golgi apparatus is it does go back and forth, but it's not quite in and out as that endoplasmic reticulum is really like, woo! And that's so it can store more stuff. These guys are a little bit more organized, which is good, because if you went to a post office, that would not be helpful. All right, so we're going to put this right next to our endoplasmic reticulum. And I'm just going to do it sort of as a series of like these little tubes that are going to be able to shoot our packages all around the cell. What mine's going to look like. It's a little smaller. Take a little artistic license. Ooh. All right. Oh, that was still not small enough. You can have another one.
What's your favorite part of the cell?
My part? Probably this DNA storybook. I just love that, like, DNA is so small but also so powerful. You know, it's like it tells me to have brown eyes and brown hair, and it tells me to look the way I am. It told me to be 5'4". Like, I think that's really cool. Like, it's so powerful. And then it also tells my heart how to beat. Like, it's pretty amazing. And you can extract it at home and see it. Like, we're going to see DNA tomorrow. And that just, like, it makes me giddy. I love science so much. Oh, my goodness.
Bye, George.
Oh, bye, George. Wait, we didn't get to see George's.
Well, oh, can you show us your thing, George?
George is in Zoom. He's finished his already. You guys are working faster than me. Oh, my goodness. All right. There's my guilty
George
apparatus.
and Dad made something.
Oh my gosh. If you guys could see George's, it's amazing. It's on a paper plate. We've got like pipe cleaners. We've got pom-poms. We've got rubber bands. Oh my gosh. I'm so digging it. There's eyeballs. There's eyeballs. Oh my gosh. They use googly eyes. George, I love it. That's amazing. All right. So I have my Golgi apparatus. I got my mitochondria. We do have vacuoles on this that are going to store stuff. So I could put some vacuoles on here. What could I use to store things?
Oh, Rayden, do you want to show us something?
All right. So I'm going to use these marbles as like storage bubbles. These are going to be the vacuoles that are on the handout, but they're not actually on the handout because
Rayden does have something.
They are. I forgot to label them, sadly. All right. So I'll put some vacuoles in here.
Oh, Calero's here.
And then the other thing that I need is sort of like a cytoplasm on this. And that cytoplasm would be some sort of like gelatinous liquid that I'd fill it with. Obviously, I don't have that. And if I put like gelatin in this, that would be hard. Maybe I could like sort of, I think I'm just going to leave it without the cytoplasm so that we can sort of see into it. So this is going to be my cell. I've got my cell wall. Hang on, Rayden, I see you in Zoom. I got my cell wall. I've got my mitochondria. And I remembered that because I'm hungry and I want to eat these noodles to give me energy. And these noodles are part of my cell and they're actually eating nutrients to make energy for our cell. I've got my little makers that are my little ribosomes, and they kind of actually stand up like little people. I kind of love it. So they're going to make things and they go to my little endoplasmic reticulum to grab all of the stuff they need. And when they're done, they're going to ship it out through my post office of tubes that are going to shoot it out. And when they need to know how to make something, they're going to go into my nucleus. And they're going to go through these little pores and they can actually get to the DNA storybook there. And the ribosomes are going to be made in my nucleolus, which is pink, which really, I'm glad I made that pink because that really helps me remember that that's where they're made. All right. So this is our cell model for today. We're going to head on over to Zoom in just a moment so that you guys can see and share all of the projects. I know Rayden is over there ready to share with us. I'm super excited to see his. Please like and share all of our stuff with people. The more, the merrier. We are going to extract DNA tomorrow. So don't forget, if you want to be ready and on time and do it with me, you need to take some alcohol or some like hand sanitizer. I think should work. I've never done it with hand sanitizer, but
vodka?
It's mostly alcohol.
Vodka.
No, actually, a drinking alcohol would not work because a drinking alcohol like vodka is 40% alcohol, and your hand sanitizer is 70%. So it's a much higher percentage. But put it in the freezer so it will chill. It won't actually freeze. And that we can use tomorrow along with some dish soap, some bananas, some Ziploc, some salt, a sieve. And you'll have that. If you're on Patreon, you'll actually have that download of what we are doing on Friday.
Again, put it in the freezer.
Oh, yes. Again, Evan's telling me to remind you. Put this guy in the freezer. Tell your parents to put some in the freezer so we can extract DNA together tomorrow. And then on Friday, we're going to make our beating heart card. Which will be really fun.
Come over to the Zoom.
All right, yes, come on over to the Zoom. If you need that link, it should be on Patreon. If you're not a patron, just come support us. It'll be for our programming, totally awesome. If you can afford to support a family in addition to yourself, amazing, because that really helps us create this program and keep it running and do that community science, but like way bigger, because this is so fun. All right, so I will sign off. It was great seeing you guys on YouTube, and I will see you over in the Zoom. Have a great afternoon. Alrighty. My Zoom family. Oh, I forgot to do my superpower.
Oh, no. Maybe you're still on.
I'm still on YouTube?
Yeah.
Oh, I forgot to do my superpower, guys. Oh my gosh. I'm super excited because I had a really fun superpower. I am going to put these super spiky, I thought of this when I was getting stuff. I'm going to put these super spiky things on my cell so that things that I don't want to get into my cell, like bad guys, can't get in. They're going to be like skewered. It's going to be amazing. I'm going to put these all around my cell. And I will try to do it quickly because I know my Zoom class is like ready. And they're like, Dr. Erica, I want to show you everything. And so we're just going to flip it down.
Your own cell, you can give it whatever superpower you want to.
Yeah.
So your superpower could be to make your cell fly.
it could make it like you're maybe you make cells that can breathe underwater so that you're like
or breathe fire,
fish,
or maybe you can turn yourself into a dragon. Your cells can do anything.
That would be cool. Yeah.
So think about
maybe you make a cell that like can see a different type of light, so you can see like heat instead of seeing like a wall. You would see the heat behind the wall. That would be cool.
Oh yeah, see-through walls.
Yeah, like you'd have different vision. That would be an amazing superpower for your cell. I love that. I can't believe I almost forgot to put a superpower on because I was really excited about making this like spiky, no bad guys can get into my cell.
Raiden, what is your superpower cell gonna be?
So here is my super spiky. Anything that's bad that tries to get in is gonna get stabbed, and I'll be like, "Oh, you bacteria, put your head on a pole." It'll be like very medieval. It'll be great. Any viruses that try to get in, they'll be like, can't get in.
No coronaviruses for us.
Yes. No, nobody will get sick with this. Like you would never get sick because it would stop anything that tried to be bad and get into your cell. Yes, I have cured all illness on the planet with spaghetti sticks. Right. I had to think of a cool name for it.
You do.
I'm not sure what it would be.
Invincimus.
I don't know.
All right, I am going to close the Zoom.
Now we'll go off of YouTube and we'll be in Zoom.
All right, come on over to Zoom.
This is your
last chance to click the link.
Perfect.
And 3, 2, 1. Goodbye, everybody.
All righty, let's check out Raidens. We have to unmute everybody, I think, and you guys will have to remute yourselves. This one's still on. How do we turn that one off?
I don't know.
All right, I'm bringing you guys closer so I can see.